diffusion, first I will talk grossly diffusion, and then I will differentiate simple and facilitated diffusion also. So what is diffusion? Diffusion, like we inhale the oxygen, and that oxygen will be transported uh, first in the pulmonary arteries, sorry, pulmonary vein, and from pulmonary vein that will go into the left atrium, left ventricle, and then systemic circulation. Through systemic circulation, the oxygen will be transported throughout the cells of the bodies. How this occurs? Let's see. So at first I would like to define this diffusion. What is diffusion? So diffusion is movement of any substance, movement of any substances that might be ions or molecules. Movement of any substances, substances along their concentration gradient, that is from their higher concentration to lower concentration, lower concentration. Here the important point, with or without the help of carrier molecules, or proteins. That carrier molecules are mainly the proteins. So in is known as diffusion. This is very simple definition. Diffusion. What is it? Movement of any substances, that is ions or molecules, along their concentration gradient, not against the concentration gradient, but along their concentration gradient, that is from their higher concentration to lower concentration. The important point is that with or without the help of carrier molecules is known as diffusion. In detail I will differentiate later, but remember here mainly this simple diffusion will take place without the help of the carrier molecules, but facilitated diffusion needs these carrier molecules or carrier proteins. So let's put an example here. What is concentration gradient? Let's suppose this is, here we make two compartment, and let's suppose this is membrane that is selective permeable for particular type of the molecules or ions. molecules or ions, and here is the some solvent, and we put here few substances that is permeable to this membrane, through this membrane, or let's make it just a round circle. Let's suppose this is compartment A and this is compartment B. If this membrane is let's suppose selective permeable for substance symbolized as this circle. Let's suppose this is particular type of the substance and this is permeable through this membrane. Other component, let's suppose let's assume that at this time is impermeable through this membrane, So where, in which compartment the concentration is higher? Here is the solutes also, solvent. Let's suppose some solvent here should be present. Since we are talking about the through cell membrane, and in gaseous condition, gaseous molecules, if there is any pore in cylinders, let's suppose join two cylinder and make a pore, closed pore. So one cylinder is empty and one is filled with gas. The gas will move from the filled cylinder to the empty cylinder. So same mechanism apply here. Here is the higher concentration of this particular type of the molecule, and here is the lower concentration. So diffusion will occur from where to where? From A to B. Diffusion will occur from A to B. A to B. This will be direction of the diffusion, and this diffusion will occur till the equilibrium is reached, that is concentration is equal in both compartment. Till then this diffusion will occur. But my dear, what do you think? These molecules will only diffuse in this direction from A to B? No. The diffusion will also occur from low concentration to high concentration. Please remember, because in a particular compartment, either the compartment is fluid filled or gaseous compartment, the molecules will be in random motion. In all compartments, the molecules will be in random motion, what I call random motion. Some authorities also call as constant motion. Random motion, due to the random motion, that each molecule will move in different or separate direction, and by that random motion the diffusion of few particles will also occur from B to A, from B to A. But the point is that the net diffusion will be occur from where to where? The net diffusion will occur from A to B. A to B, that is, the net diffusion will always take place from higher concentration to lower concentration. Hence the definition is from their higher concentration to the lower concentration, but remember, the molecules or ions can move from low concentration to higher concentration also in few amount, but the net diffusion will be the from higher concentration to lower concentration. Now the question arises, this diffusion, what is the force band that cause the diffusion of the molecule, or energy that cause diffusion of the molecule? So for that you must know that this energy for diffusion will be provided by heat, that is thermal energy will be main regulatory, but the direction will be managed by direction and rate will be determined by concentration gradient, then pressure difference or pressure gradient, and pressure difference and charge difference also. What does it mean? Like if we apply here more pressure, the rate of the diffusion will be increased, and in case of this charge, let's suppose this is positively charged molecule and here is negatively charged molecule, so this will also increase the rate of diffusion through this membrane, and concentration of course, higher concentration to lower concentration. Please remember that thing also, that this pressure difference, charge difference also affect the diffusion of the particular substance, and due to this random motion the diffusion is taking place. In addition, the diffusion will be favored by the pressure difference, charge difference, and concentration difference also. Now from chemistry, from the knowledge of chemistry, you must be knowing that the molecular motion will cease only at absolute zero temperature. Molecular motion of the substance will cease only at absolute zero temperature. So this is all about the diffusion. Now I will differentiate the simple diffusion versus facilitated diffusion. simple diffusion versus facilitated diffusion. Put here simple diffusion, and here the facilitated diffusion. Diffusion, as I mentioned earlier, this diffusion will occur without help of carrier molecules. Or proteins, and this facilitated diffusion will be mediated by or helped by carrier molecules or transport proteins. Now, so what will be the pathway? Like this, let's suppose this is the, again I will do a cell membrane. And let's suppose this is channel proteins. This is channel proteins. So either the diffusion will take place through these channel proteins, Let's suppose this is inside membrane and this is outside. In reverse direction can also the diffusion will occur, outside. Either or this through this interstices of the lipid bilayer. Bilayer molecules can move by this simple diffusion mechanisms. So can occur through interstices of lipid bilayer or channel proteins. So again, please remember, if the okay, I will mention, by writing time will be consumed more. So what happens, this diffusion will be higher in case of the smaller size molecules or ions, or if that ions or molecules are the lipid soluble, that lipid soluble and a smaller diameter molecules will occur, molecule will be transported in faster rate than larger diameter and or lipid insoluble molecules. This is the condition. Here. So the facilitated diffusion how will occur? Let's suppose this is accelerated diffusion again. Let's suppose this is carrier molecules. Let's suppose this is carrier molecules. So as soon as somewhere here in that molecule will be binding site. At binding site, as soon as the substance or molecules that is to be transported will bind the conformational change will occur in this carrier proteins, and that will transport the molecule from one side to the other side. From molecule one side to the other side, the main mechanism is there should be conformational change in that particular carrier molecules. So this substance in this way will be transported from one side to the other side. So only this will only occur through carrier proteins or molecules. Carrier proteins. Now again I will differentiate these two by a graph. This simple diffusion and accelerated diffusion rate and concentration I will differ in that aspect. These two, simple diffusion, and there are many difference also minor, so please read that also. These are the major differences. Here I will draw a graph. Like let's suppose let's suppose this is the x-axis and this is y-axis. Then I will draw a straight line passing through the origin. This is one graph, and next graph, let's suppose again starts here. The moral of the story is that see the different curve of this graph. This curve is of accelerated diffusion. This green curve will be of accelerated diffusion. And this straight line will be of simple diffusion. Let's suppose this is rate of diffusion rate of diffusion, this axis, and this axis of concentration gradient. Concentration. Please notice in the graph that as the concentration is increasing, this simple diffusion, the rate of simple diffusion is also increasing in linear direction. According to this concentration gradient, and this accelerated diffusion, in case of this accelerated diffusion, the rate at some point, the rate initially concentration gradient, increasing the concentration gradient, let's suppose this is concentration gradient axis. As the concentration gradient increasing initially, the facilitated diffusion, the rate is also increasing, but at certain point, this rate is plateaued, like I forgot the spelling. Please remember this plateau. Why occurring here plateau? Let's suppose this is the like this is the Vmax, maximum velocity of diffusion of this facilitated diffusion. But in case of simple diffusion, as long as you are increasing or creating the concentration gradient, the rate is increasing. But somewhere in facilitated diffusion, after reaching the maximum velocity, maximum rate of diffusion, it gets plateaued. That after this Bmax, if you increase the concentration gradient further and further, the velocity, the rate will not increase for this facilitated diffusion. Why this happen? Let's see. As I mentioned, this simple diffusion will not need the carrier molecules, but this needs carrier molecules. My dear, these carrier molecules, for particular time, carrier molecules capacity is limited. Why? Limited because for constant for a particular time, its carrier molecules number is constant. Let's suppose number is constant. As the concentration gradient you will increase, it is not like that this carrier molecules number will increase drastically. Not like that. Number is constant, and its capacity of the transporting, that what will happen in carrier molecules, that let's suppose this is carrier proteins, here will bind a molecule, and from here it will be released. Like then binding and releasing and rate of conformational change is in limited capacity. Like everything has the capacity limited. Beyond this capacity, also even you increase this concentration gradient, this factor will not let them to increase the rate, let them to increase the rate of this facilitated diffusion. So this is the postulated mechanism that this conformational change, loading and unloading, is limited capacity, and carrier molecules are also in limited number. So this will limit its capacity, hence the facilitated diffusion after reaching the maximum velocity will not increase further even by increasing the concentration gradient. So after mentioning the difference, please make this graph, you will be getting good marks. So these are about the diffusion. Now move on to the osmosis. Oh, okay, I forgot to mention the examples of this different: simple diffusion, facilitated diffusion. Examples will be here. Examples is diffusion of, as I mentioned earlier, diffusion of oxygen O2, diffusion of oxygen from long alveoli to pulmonary capillaries, that will drain into the pulmonary vein, capillaries, to the pulmonary capillaries, and diffusion of carbon dioxide in opposite direction, that is from pulmonary capillaries to the alveoli of the lung, and then will be exhaled out through the respiratory tract. And also here you can mention the examples like same this diffusion of the oxygen from the systemic capillaries to the tissues, and from tissues or cells, the carbon dioxide will diffuse into the that systemic capillaries, and that CO2 will be carried by the venous blood, lung, then out through the respiratory tract. So this diffusion occur by the simple diffusion mechanism. And facilitated diffusion, there are many examples of this facilitated diffusion. For example, like different glucose transporter, different glucose transporter, like there are till now there are 14 different glucose transporters have been identified, and there might be many more too. So for example, GLUT GLUT4. This GLUT4 is important transporter. GLUT4 is important transporter that transports glucose in skeletal muscle and adipocytes, and diffusion this diffusion will be mediated by or enhanced by the insulin. Enhanced by rate will be enhanced by rate of diffusion will be enhanced or increased by insulin, because these are insulin sensitive tissues, enhanced by insulin. And some authorities believes this insulin will increase the rate of diffusion by 10 to 20 times than normal. Like here again what can be a MCQ question in endocrine block, that the glucose uptake is dependent on insulin by this skeletal muscle and adipocytes only. In other tissues, the diffusion of the glucose or uptake of the glucose will be irrespective of the insulin. That is there no need to the insulin to be present for that uptake, like in brain, in kidney, in cardiac muscle. In that cases, the glucose is uptaken in absence of insulin or irrespective of the insulin presence or absence. So these are the accelerated diffusion, and like GLUT2 is found in the pancreas, beta cells of the pancreas. There also the glucose entry occur and at certain condition, and then the insulin will be released, synthesized and released. So this mechanism I will talk in detail about this mechanism in endocrine block. So these are the examples of facilitated diffusion. Like galactose and fructose will also be uptaken by this GLUT transporter, and that all will be the part of the facilitated diffusion.